Hi, everyone. Sorry, you went from my last points to now hearing more of my voice. Apologies. So as mentioned, I'm standing in for Viantha, who very kindly shared her slides with me. So I will try to stick to them. Sort of was asked to step in to speak about training. And as I was discussing with Lisa earlier, I can probably speak about this for hours. So I'm going to try keep it. Tell me when to stop, but I'll try keep it. I just want to clear a little window that's popping up on my screen. There we go. That's good enough. Viantha structured this specifically in the context of Confocal, which I think is useful to try to rein me in. But one of the challenges with training is it helps to be trained with something that you have access to. So in the context of South Africa, if there are not a lot of systems, not a lot of institutions with systems with reference to Lisa's sort of session, it can be difficult to get access and to get training, all the typical challenges with that in terms of access being limited by staff, by system downtime. And what we find within our facility is actually staff capacity. Demand far exceeds the capacity that we have in terms of our time to actually train users well to be able to independently work on the systems to a standard where they're getting interpretable data as opposed to pretty pictures. There's the standard sustainability challenges with regards to this, which also links back to questions about cost recovery, funding models, sustainability of staff's time. We have found one of the biggest challenges is recruiting appropriately skilled staff that doesn't have a massive ramp up stage to where they are able to independently train users, especially as the systems get more and more complicated. So let's see. In terms of, and this is, I guess, a question that I'm going to throw out. I'm going to try adopt the same model of not just prattling on for too long. Viantha's got points there in terms of there being workshops, which I know Lisa runs here in Stellenbosch. We started running them on an annual basis, at least in terms of fundamentals at UCT. But on the whole, if we sort of put it out there, what seems to be the balance ratio between the demand for workshops versus one-on-one, tell me how to use a microscope training? Is there a sense within the community? Because those are technically two different types of training. I don't know if anyone online wants to make a comment with regards to that, or Ben. We're just wrangling microphones here in the room.
So I think that's actually a very important point. And from my experience in my group, I make always a conscious effort to offer to every single student the whole single person training. But of course, you don't know in the beginning whether that person is apt for it and would like it. But I deliberately start at honors level with that, so that if the person is into it at masters, some techniques can already be as good as, and then at PhD, really at a good level. And so, but there is really that mix between group training versus concerted single person driving. And it links a little bit to, because some of those that are really good are now in imaging facilities elsewhere outside of South Africa. So that is really an important consideration. And how do we cater for that best, right?
And I guess linked with that, the two aren't mutually exclusive, because often you're able to go in depth in the theory, for example, much more in a workshop and cover all the background that otherwise never doesn't get covered.
That is exactly my experience, is that we mix the two. When I start with the student who's one-to-one, I realize they don't understand the PSF and that, and I'm not going to spend their booking time to explain the PSF. So for those, often the majority of independent users have at some point attended a proper confocal course with us. Our confocal courses is also sitting on the microscope. This is this button, that is that button. Can you see how it changes if you change the spatial resolution in pixels, etc.? So there is instrument training as a small group as well, which saves the staff time, because if every time you have to, especially in COVID time, I had to train person by person, and to explain every button to one by one, it's really annoying. So that's where I find it's both are equally, it's a 50-50. Everyone wants both. They want single training, one-to-one, and they want to do the course for their understanding.
Do you find that users can after doing that course, sitting with them at the microscope, but on a group, this is this button, that button, etc. Do they still need additional hands-on training before independent use?
Yes, because it's 2 days of which 2/3 are instrument training and 1/3 theory. But the quickness of them being able to image by themselves is really worthwhile. I mean, it takes a lot quicker for someone to train when they've done the course than the opposite. So, because of course there's not always a course. So sometimes I train one-on-one first. I wanted to say something else on that. What was it? I'll think and answer that.
But my question as well is, I mean, normally, especially for microscopy, is if you have training, how quickly do you actually use the microscope after that? Because, you know, it's fun to have training in the beginning of the year, but then you only use it in August and you've already forgotten what you have to do. So that's also the...
Yeah. Oh, that's excellent synchrony. Well done. Let's see if there's anyone online. You see? No, go ahead.
Yes, a person who gets training in February can't use the microscope that quickly in August. It's basically training from the beginning, but there's benefit to it, to train someone before they even start their imaging, because a lot of planning goes into the imaging. So I get half of the course comes only in August, but then they've planned their experiment much better than someone who turned up and said, this is what I have, and I have to train them. What is the controls? You can't put this together with that, etc. So there's benefit to both of them. They can do it for early on, plan better, but get proper instrument training when they come in August. Or I've done the course now, you know my samples already. Next week I start working 2 hours and they
How often
independent.
do you run the course?
I used to do it 6 times a year. The first half of the year I spend on that, like until about July. And then I tone down. Sometimes I have to organize another one or maybe 2, but it gets very hectic the last half of the year. Yeah.
I'm genuinely impressed. That sounds brilliant. I might chat with you and try to adopt that. Does anyone online want to say anything? I mean, Lisa and I are not too far from each other geographically, but further afield. Aurelie, are you still with us and want to add anything, or have you had to leave?
I think it looks like she had to leave.
Adrian? Adrian, I can see your hand up. Go ahead.
Thanks very much. I think it's even doubly more difficult if you don't have a dedicated person running the facility. So, you know, for example, at Rhodes, we have the microscope, and then the training is essentially done by either myself or Dirk often will come and do some training, but then it tends to be very sporadic. And if you've got a wide range of different types of analyses you have to do, then, you know, it becomes really, really challenging. And so I think we've benefited a lot from the courses, but there's no real replacement for, you know, time on the microscope and doing it yourself. So I don't know how, short of getting a dedicated person, you know, we can really streamline our training, because it's a major barrier in terms of time.
Yeah, I can completely understand that, because it's a huge barrier, even if you are a dedicated person. So I can understand that. And I think that's maybe something with continued thought and discussion as a community in terms of what can be done. So I think just in terms of trying to keep the conversation moving, I'm conscious of time. I think one thing that's come out clear is there are different modes of training and all of them are useful. And ideally, you would want to be able to do the group course where you've got a workshop going deep dive into the theory, maybe group trainings that sounds much more efficient on like the starter orientation on the confocal, and then the one-on-one deep dive, probably as your samples are ready, in acknowledgement, Chantal, of what you said, because we see the same thing. If there's more than about a 2-week gap between the training session and when people actually start imaging with their samples, they've forgotten everything. Oh, Sine, do you want to jump in?
Yes. Hi, can you hear me?
Yes.
All right, I'll be short. I'm also cognizant and mindful of everybody's time, but I was just thinking to myself, if and listening to Lisa, how you do the training with a certain amount of theory before you can actually move on to practical. What would the value be of having some of local produced content recordings on the basic principles of certain things, basic sample prep and so on? I understand that there's a theory component to it as well, but if your users have are planning at least preparing research projects and so on, then they might find quite a bit of value of watching a recording before they can actually come for a theory or book a practical session. You can save some time because these theory things they can work through at their own time and leisure. And I don't know how that structure would be, but it might be valuable to have a collective on the SABI website or whatever to refer people to say certain things you can do online at your own time, and then you can book practical sessions to optimize the time that's spent on training as well. It could also be shared resources and so on. I'm just thinking we have a compendium of resource and training information on a Zeiss portal, for example, that is shared either to customers or so, and people can watch this at their own time. And this also saves time for us when we have to do practicals. Right.
So now I love that idea. We've Mike and I have discussed that a lot in terms of what could be done to, as you say, kind of get people not starting from zero when we sit with them. We just haven't had a chance to implement it. So leveraging what Zeiss has available, and there's other resources out there as well. I think maybe again as a community is lowers the hanging fruit a little bit versus everyone doing it themselves. I think it's a great idea. I'm just going to go on. So I know Vianta had sort of prepared a table of sort of strengths and challenges, which I think a lot of this has already been touched on. There's a spectrum of systems that we have available, but they're more challenging. I think maybe in the interest of time, I'm actually going to jump to the next slide, because I think it's more interesting to keep the discussion going. And something that I'm interested in touching on is the question of in our training, because already we all just, in speaking about it, we've spoken about the theory and at the microscope, but the microscope alone does not an experiment make. There's sample, there's the data analysis afterwards, there's the techniques depending on what you're trying to do at the microscope. And I guess a question to everyone is what the sense is in terms of how well we're doing to train appropriately or get everyone working at the appropriate level across that spectrum.
If I might go on. So yes, I agree that I think we as microscopists miss sometimes the prep part that comes with it. And I think it doesn't get relayed to students how important it is or how long it takes. A few times now, there's students that come this last few months of their thesis. I have X amount of samples that I have to run, or this is my microscopy that I have to do. And then you sit there and think, this is not possible in the amount of time that you are left with. So not only the prep, but it's the imaging, it's the data analysis afterwards and getting proper data after that. It's, for me at least, the people I deal with, seems like the microscopy part is normally the last thing they want to sort of think about, because they think all the other lab-related essays and those takes long, but they forget how long microscopy actually also takes. So yeah,
It's I see this question line, but I'll just want, it's not just the students, and it's not even just the supervisors. It's it seems to be, and maybe this comes back to something promoting an understanding and awareness of microscopy and how long it takes, journals and reviewers as well. I've more than once have had to sit down with a PI and a postdoc who said, we've got reviews, we've been given 3 weeks to do this, and they've asked us to redo and add on all of this imaging that our PhD student who's now somewhere else did. And I just look at them and go, even if I gave you all of my time for the next 3 weeks, you're not going to get this done. You don't know how to do the sample prep. You're going to have to optimize it for your hands. Then we're going to sit at the microscope and we're going to make, like, it's just not possible. So yeah, online, Devesh, me? Devesha?
Hi, Devesha, yes. I just wanted to talk about something very similar to what's been discussed. Often microscopy is the last part of a study, and it's just a confirmation of this larger part of the study. So in terms of how to make this, this training, these training initiatives more inclusive, could there not be something where it's not as advanced, where it's a simple kind of a summarized version of how to get what you want, but maybe not in the detail that a person who's doing a study that's quite focused on certain antibodies or proteins that would have to go more deep into the study. So I mean, it does of course depend on the research question. So if you look at these the sample prep and basic techniques, maybe stop it there, where they don't need to go much further into it. Maybe like a kind of like a brief course or something. I don't know.
I think it's a really good idea. And while the microphone gets handed over to Ben, I'll add my response. I think it's appropriate to do material and training scale to what people need. I will say we ran, I think it was a 5-day course on sample prep earlier this year at UCT, and it covered basic histopathology and staining and basic immunofluorescence. I think it was on tissue and on cells. So yeah, it covered a range of samples, but that alone took 5 days. There's what we do is not straightforward, and I think it's easy to forget that. Ben, what do you want to say?
Yes, yeah. So if I think of what from the community side, what I feel is really critical, especially as now the network is starting to establish and the students are geared towards the imaging, is really, I think, the basics. So the basic techniques and the sample prep. So the first 2 points, I think, would be for me the first thing to really, with recordings, as Soleil indicated, and maybe some kind of standardization across our groups. I think that would be very, very powerful. And then I think, if I think of advanced techniques, we kind of failed there also a little bit from our experience. We can see it often if we focus, let's say we focus on FRET, we do some dedicated training, then projects will emerge around that. But then a few years, we don't do any of that. So I think it will be helpful to have also some dedicated plan for maybe also through SABI, that we say, okay, 2026, we focus on X as the advanced technique, plus the basics. But the most important at the moment, I think, is sample prep and basics, because there's already so much that's needed, really. And the students are also so eager to jump quickly to the fanciest method that they have seen, and that we first say, okay, yes, very good, but this first needs to be
Agreed. Devesha, do you want to add to that? Your hand's still up. Is it still up or up again?
Sorry, it's not up.
Yeah, Ben, I agree. And I think a communal approach to this is a great idea, because we have had a strong focus on teaching fundamentals the last few years, because we've seen that that's what's needed. But I would love an excuse to teach someone super res again. Yeah, and to cover FRET, to cover FCS, I think there's the hyper or spectral imaging course planned for next year at UCT. Multispectral, that's a thank you. And agreed, just because we have a need for the foundational stuff, and not to neglect that, there's scope for also bringing some attention to the advanced stuff. And I think we've kind of already touched on image analysis, but I don't want to neglect that. I know it's something that we struggle to do sufficient training on for all the reasons already discussed, but I don't know if anyone wants to add anything in this regards.
I think the image analysis is definitely as important. I feel it's just as important as the sample prep and basic microscopy, because that's the bottleneck. And it's the part where the staff imaging scientists cannot dedicate more time on. What I think is necessary in SABI is that we start identifying who are those people. I know of 2 people I can ask for image analysis support, Renzo Tiard and Nicola in our department. Otherwise, I have no idea who else. And I think it would be very valuable for SABI to start identifying who are the people who really have skill, and motivate those people to also bring other people into the network. For example, Renzo Tiard's students. Maybe we should really have a discussion with him, and whoever is at UCT or UP or wherever, where we can find out who are those people we can also draw into SABI, because it's bioimaging. So if they do bioimaging analysis, we can draw them in. They don't have to have a microscope, I think.
Completely agree. With the last, we're already over time, but last minute, because I think this is a huge need, especially amongst the core facility people, is the training on maintenance and calibration. Who's actually been trained to do this? Chris, you don't count. You work for Zeiss.
Basic training.
Basic training. I've had the opportunity of going to Jena and being trained by Zeiss. It was fantastic. It's still hard to make the time and to remember to do it. Is this something that can also be, and I think given the 2 Zeiss people in the room, I guess I'm speaking to both of you, do we have to fly everyone to Jena, or can something be organized locally in terms of not just help, our microscope's not working, can you send one of your 4 engineers to us, or help troubleshoot? Sine, jump in.
Yeah, so I have some good news for you. We are working, and I will not commit to a timeline in an open forum, but hopefully
Oh, come on,
we
Sine.
we will be soon. Soon is a very vague concept in South Africa, but hopefully soon we would be able to host train the trainer programs to train technical staff working in core facilities locally, and also certify them. So this is something that we've identified, of course, over the last 2 years. It's increasing, and we do see the value in bringing these courses locally. And it will be dedicated to core facility people or advanced imaging specialists who have to take ownership of instruments to be able to support at least some basic troubleshooting and maintenance and calibration. Okay. But the dates will be communicated hopefully next year. All right.
That's super exciting. Thank you, Sine. I look forward to finding out when soon is. The last question is on impact. I think we've had some really good suggestions on what we can do as a community, which I think is a great way to bring impact to this, and I think also sustainability. I don't feel the need to be the last person speaking, so I'm going to throw the question out to everyone one more time. Otherwise, I'm going to ask who I can hand the microphone over to. We're just doing a microphone transfer here.
Thank you, Caron. So I think that reminds me of course, impact, impact for whom, right? That we have so many avenues to cater for. So I think we should carefully think about what kind of impact, whom do we want to reach, right? So on the one hand, of course, impact to our VCs. That would mean student graduation and papers. High visibility impact papers would be there appropriate. Exactly, yes. And so there are so many avenues, right? Whilst impact may come in very different aspects. It may come in really good visibility of community training or awareness. It might be the NRF and DSI is again other metrics. And so I think we should try and explore through SABI what areas we want to hit when, and then do that strategically. I think we have all the tools for it, but we are not strategically actually doing it. And it of course, at the EOE meeting, right, the discussion around impact, there's so much impact that we have, but we have to kind of allocate it so that we do it effectively. Because after all, I think SABI, the impact has to reach the decision makers. And that is actually above the VCs. If we think of, again, the NEP and the MRC and the NRF and the road mapping, that's where the money flows. And hence we need to reach also those levels through what we do. So it's a really important aspect. And I think it deserves a topic on its own and a strategy to go about it, like we do now with the SABI outlets and the LinkedIn and so forth.
Hi, Ben. Yeah, I want to I also want to just comment on what you said. I think in a micro, I think from a micro perspective, not microscopy, just from a beginning perspective or a simplistic perspective, I think what maybe could help is that SABI initiates a core facility manager planning meeting, for instance, for the year. At the beginning of the year, where all the core facility managers come together, discuss what they want to do and what they want to plan, and then in that way support the greater microscopy community. And if there's then a topic like super resolution or multispectral or FCS or all these other advanced techniques, that there's that everybody sort of agrees with the direction that the core facilities in South Africa wants to take, and that shows unity amongst the core facility managers, and everybody then also knows what's going to happen for the year moving forward. And that way you can strategize around that, and you can sort of direct your PIs or the PIs in the universities that you have direct contact with in a certain direction, knowing what's coming and what is being planned and what the direction is for South Africa as a whole within the research sphere. Mike?
Hi, everyone online. I apologize. I have just joined this meeting, having been in a meeting about training, but I thought I would share some thoughts because apparently I like the sound of my own voice. But the kind of training microscopy has to go, the technology of microscopy also has to be always viewed in the context of the biology. And you can never say that the technology drives the biology, and you can never say the biology drives the technology. There's two sides to that coin, and that's what I think makes it difficult, because fundamental training of what can be done with a microscope, how does a microscope work, that stuff is broad and kind of generalizable enough that you can dish it out in workshops, you can dish it out in courses and things like that. But the more advanced you get, it needs to be done hand in hand with the biology. And that, I think, is a particularly difficult thing to kind of reconcile, because at the end of the day, this entire thing of whether we're talking core facilities or microscopy labs and things like that, it gets judged on its success. But the success is, I believe, currently very misaligned, because in our expectation, for example, we're only based on what research papers we get, not how many people we train. But you can't get people using the systems until they're trained. So there's this horrible misalignment of, we're jumping the gun. The expectation is far too much right now. But even if we could do all the training we wanted to do, where would we focus our efforts? Because if we're talking about sustainability as a primary thing, how do we make it sustainable? We can't train everyone about everything all the time. How do we pick where we allocate our resources? And I don't think there is one answer to address every eventuality. I think it's a combination of workshops. It's a combination of potentially a postgraduate degree. It's a combination of online tutorials. It's a combination of hands-on training. It's a combination of going to Jena. How we structure that into a coherent thing that is easily accessible is a challenging thing.
I think, Mike, for someone who's just walked in the room, you did a great job of pulling on a lot of threads of like what has been discussed. No, it's a great, not in a unnecessarily repetitive way, but a great way of pulling themes together as a final comment. So thank you. Good job. I think one thing that has come out and basically from the two ZEISS reps that I think is a fantastic suggestion is at some sort of annual, if possible, in-person meeting. I love the idea of replicating the core facility managers RMS or UK Bioimaging, I'm not sure which one it is, sort of annual meeting, and using that to coordinate. Because I think that's one way you make it sustainable and make sure the necessary bases are covered and where we want to go can be covered as well. And I think doing this as a community way to find sustainability. It's a promised land. I'm not sure we're there yet, but we can get there.